Today we will be talking about the cell cycle checkpoints in cancer. Here's a diagram that shows the whole cell cycle. First, we begin with interphase. The cell spends about 90% of its time in this process. Here the cell's chromosomes duplicate, and we can clearly see that there is a nucleus in the cell. These lines represent microtubules, and these yellow-green thingies represent centrosomes. Next, mitosis begins and we enter the prophase. Here we can see that the chromatid pairs are now visible and the spindle is beginning to form. We can also see that the nuclear membrane is starting to break down in order for the cell to divide. Then we move on to the lesser-known prometaphase. Here the spindle microtubules are attached to the kinetochore. After this, we move on to the metaphase, where the chromatid pairs are lined up in the center of the cell. Next is the anaphase, in which the chromatids separate, which is shown by the X's becoming V's. Then we move on to telophase, where cytokinesis, or the separation of the cytoplasm, is taking place. Also, the nuclear envelope reforms so that each new cell will have a place for DNA to be stored. These new cells move on to the interphase to live out the rest of their cell life and perform their jobs needed for our body to function.
This is where things get a little complicated. The interphase consists of three phases: the G1 phase, the S phase, and the G2 phase. During the G1 and G2 phases, the cell is growing. The monkey in the middle of the three, which is the S phase, is where the cell will duplicate its DNA. Again, this is where things get a little complicated because a lot of people confuse the phases of interphase with the checkpoints of interphase. In the G1 checkpoint, the cell will continue its daily functions and it will increase in size. Also, it ensures that there is no damage to the DNA and no growth factors are affecting the cell. If the cell does not pass the G1 checkpoint, it will then move on to the G0 resting stage. Here, it will fix its problems until it is able to pass the G1 checkpoint. At the G2 checkpoint, the cell is checked for the appropriate cell size and that the cell is healthy enough to reproduce. If the cell is not the adequate size, it will continue to grow until it gets big enough. If the cell still does not pass, enzymes like cyclin, which we will go on to later, will fix its mistakes in the DNA. After the G2 checkpoint, we move on to the M check, which is also known as the spindle fiber checkpoint. Here the cells are checked to make sure that all of the chromosomes are attached to spindle fibers and ensure that the cell will divide evenly between the daughter cells. This is the final go signal before the cell enters anaphase. Once the cell enters anaphase, it cannot stop the process and it is irreversible, which is why it is crucial for the cell to have checkpoints during interphase. In the diagram to the far left, the green part of the, peanut-shaped thingy represents cyclin, which is an enzymatic protein. The red part is a CDK, which stands for cyclin-dependent kinase. The CDK adds a phosphate to the cyclin, which starts a reaction for the cell to be able to do numerous jobs in the cell cycle. CDK, along with cyclin, is a major control switch for the cell cycle and will allow the cell to process through the cell cycle. On the top graph, we can see the different concentrations of cyclin, depending on the phase in which the cell is at. We can see here that cyclin D is present throughout the cycle and gradually has a sort of semicircular shape to it. Cyclin E starts in the middle of the G1 phase, peaks during the transition from G1 to S, and is over by the middle of the S phase. Cyclin A gradually increases from the G1 phase through the S phase and reaches its peak towards the middle of the G2 phase. Then it declines and is done about a quarter of the way through mitosis. Lastly, cyclin B starts steadily increasing at the beginning of the S phase and it reaches its highest point during the very beginning of mitosis, and then rapidly decreases, and then towards the middle of mitosis it ends. Next, we have a graph that shows the MPF activity and cyclin concentrations during certain phases of the cell cycle. The G1, S, and G2 phases are a part of the interphase, while the yellow arrow represents mitosis. We see from the graph that the MPF activity and the cyclin concentrations, We see from the graph that the MPF, or maturation promoting factor, activity spikes during mitosis. MPF includes the CDK and cyclins and trigger progressions through the cell. And the resulting MPF complex, when associated with these cyclin-dependent kinases, phosphorylates a variety of proteins and initiates mitosis. So here we have a PDGF, which is a platelet-derived growth factor, and it's one of the numerous growth factors for a protein that will regulate cell growth division in the cell cycle. In particular, it plays a significant role in blood vessel formation, or angiogenesis, the growth of blood vessels from already existing blood vessel tissue. So here we have this little experiment with the tissues in the Petri dish, and the scalpels are cutting them up, but then they are suspended when they are broken down by proteins that were enzymes. And then here you can see that without the PDGF, the solution is still bluish, and with it, it is pink, so you can see that the growth is occurring there. Sadly, we all know about cancer, whether we have a family member that had it or we just heard about it on TV or Grey's Anatomy. Cancer is caused by an uncontrolled cell growth and the failure to regulate in the cell cycle, where it's just an abnormal growth of cells. This means that your own cells grow out of control. Different cells have different mitosis rates. For example, hair follicle cells have a high mitosis rate, which is why hair grows very fast. On the contrary, your brain and nerve cells don't go through mitosis very often, which is why when you lose nerve cells from a crash or particular life choices, you can't get them back because they do not replicate. These cancer cells appear and then just divide and divide and divide until they form a tumor, which is just a mass of cancer cells. Typically, when somebody has cancer, one treatment they will do is chemotherapy. Chemotherapy works by targeting cells that go through mitosis quickly and divide quickly and killing them off, which is why those going through chemotherapy tend to lose their hair. Here in the diagram, we can see that the normal cells are fine and everything's good, and then these abnormal cells begin to start, and they begin to multiply and multiply until a tumor forms, which is shown in this one. Okay, that's it. Thank you for listening, and yeah, yay!